Thank you very much, Daniela. Thank you very much, Pietro, for inviting me here. It's an honor to be here to give this presentation in such a prestigious hall and to represent Italian neuroscience here at this prestigious meeting. So you've heard the talk, and the title of the talk, and let me briefly introduce you to who Lombroso was. He was a physician who was also a criminologist, and he insisted he had a theory of anthropological criminology, which essentially stated that criminality was inherited, and that someone born criminal could be identified by physical defects which confirmed a criminal as a savage. So this physical information that he was trying to gather from which to understand whether a person was anthropologically criminal was especially some facial features. Of course, there isn't much to be proud about the theory of Lombroso in the sense that it gave birth or it participated to some of the eugenics that followed afterward in the following century. But what it was very clear to him, it was that there were some human behaviors that are heritable or inherited. And following on this line of thinking, what we have been studying in my laboratory in the past few years are these two main lines of research. One is to try and understand the genetic and environmental risk for schizophrenia, and the other is how can we identify some biological pathways which are prone to gather genetic risk for schizophrenia. So let's talk about the first line of research first. As many of you know, many psychiatric disorders are largely heritable, and these disorders include schizophrenia. This is information treated by a review paper by Sullivan, and you see that disorders like schizophrenia, autism spectrum disorders, and bipolar disorder are highly heritable, meaning that around 80% of the variants of the phenotype is explained by genetic variation. But genetics is not everything for many common disorders. There are also environmental risk factors which are recognized to be risk factors for schizophrenia, for example. One of these risk factors is obstetric complications, and another one, which has also been recently established, is being victimized, being bullied by other people during adolescence. And in the search for schizophrenia genes, up until a few years ago, several labs in the world concentrated on the so-called hypothesis-driven genes, or single gene approaches, and our lab focused on the dopaminergic pathways, especially the dopamine D2 pathway, which has been implicated in the pathophysiology of schizophrenia for decades based on a treatment with antipsychotics, which are dopamine D2 blockers. And then this landmark paper came out a few years ago now. This is the first wave of the second wave, actually, of the Psychiatric Genomics Consortium, which gathered a large number of subjects and performed the so-called genome-wide association study. And this was the first study that unequivocally established that there are 108 genetic loci associated with diagnosis of schizophrenia. Now, without going into much details about this, time is limited. A few of the key points of this paper were that many of the genes involved by this study were not genes that we could have known before, and only a few of them actually made sense from the neuroscience that we knew before, like, for example, the dopamine receptor D2. But most of the genetic variation that has been associated with diagnosis of schizophrenia by this study is intergenic, and it seems to serve a regulatory purpose in the translation of DNA information into mRNA and protein afterwards. But one of the also important points that this paper scored is that now we can have genetic information about each single individual, in which we can calculate a polygenic risk score. Once we know all the genes, or at least many of the genes that are involved in genetic risk for schizophrenia, based on the allelic information present in each individual person, we can calculate for a single person a polygenic risk score. And with this polygenic risk score, we can summarize what the genetic risk of that single individual is for schizophrenia. What is the advantage of this? First advantage, and most obvious advantage, is that we can calculate the interaction with environmental risk factors. And this is what Danny Weinberger and Gianluca Orsini did in this paper published in Nature Medicine, in which we also collaborated, in which the following was demonstrated. First of all, as you can see on the graph on the left, of course, the polygenic risk score, which again summarizes the genetic risk for schizophrenia for each individual, of course, as you would expect, is greater in patients with schizophrenia than in healthy subjects. But when dividing up the subjects based on whether they had undergone early life complications like obstetric complications, you can see that the results change quite significantly, in the sense that between patients and controls with no early life complications there is no significant difference, whereas there is a statistical interaction between genetic risk for schizophrenia, diagnosis, and early life complications, in the sense that patients with schizophrenia have greater polygenic risk score in the presence of early life complications, suggesting an interaction between an environmental risk factor, which was already known, and the polygenic risk score in patients with schizophrenia. And of course, the greater is the early life complications, as summarized with this score here, the greater you see the difference between patients and healthy controls. Also, with the polygenic risk score, we have summarized the whole genomic risk for schizophrenia. But what are the genes that do provide an interaction with the early life complications, like obstetric complications? And we can actually see it in this graph. A placental polygenic risk score was calculated versus a non-placental polygenic risk score. Basically, a polygenic risk score based on genes expressed in placenta versus genes not expressed in placenta. And you see that the bulk of the significant difference is found in the polygenic risk score for which the genes are expressed in placenta. So suggesting that it is the placenta mediating this interaction between obstetric complication and genetic risk score for schizophrenia. And ontogenetic analysis also showed that response to stress and genes responding to hypoxia are the genes that are involved in this complication, in this interaction. Also, in our lab, Giulio Pergola also performed another study between polygenic risk score for schizophrenia and another known risk factor, environmental risk factor, which is being victimized and being bullied. Giulio opened up a collaboration with a group in the Netherlands, which had studied a group of around 600 kids, followed up longitudinally over 3 age periods of time: 10 to 12 years old, 12 to 14 years old, and 14 to 16 years old. Just remind you that the peak of age at which there is greater risk for schizophrenia and symptoms start to appear is between 14 and 16 years of age. And what Giulio has shown here is that genetic risk for schizophrenia interacts with being bullied by other people only when at a specific age range, which is between 12 and 14 years old, not before and not after, and that this interaction, as evaluated with a mediation analysis between genetic risk for schizophrenia, indirectly predicts the frequency of psychotic experiences at 16 years of age, and the mediation effect is performed by bullying victimization, suggesting again a specific way of how genomic risk interacts with environmental stress factors. So now that I've shown you a few examples of genetic and environmental risk for schizophrenia and how they interact, we can see how we can try and use different kinds of information to understand how genomic risk for schizophrenia may involve different biological pathways of risk for schizophrenia. So we have used the following approach. We have used the transcriptome-wide information from post-mortem brains in collaboration with the Lieber Institute for Brain Development, for example, in order to study the whole transcriptome, the whole human transcriptome. And we want to, again, here the attempt is to try and characterize biological pathways which are involved by genomic risk for schizophrenia. So in order to categorize the information in the whole transcriptome of the human brain, we use co-expression analysis with this software which is called WGCNA, Whole Genome Co-Expression Network Analysis, because the idea, one basic tenet of biology is that genes are co-expressed together. Large groups of genes are co-expressed together because they are co-regulated, namely identifying specific biological pathways by means of how these genes are co-regulated and co-expressed together. So by analyzing co-expression analysis, we hoped and we think we have succeeded in identifying specific biological pathways which are completely data-driven. No hypothesis is involved in this. Of course, once we identify this network information, this co-expression information, we want to try and translate this genomic risk for schizophrenia into in vivo information, because we want to understand what's happening in vivo with the belief and the hypothesis that the genomic information is shows up in different phenotypes that then lead to the phenotype of schizophrenia. These different phenotypes, for example, being functional brain imaging, with which we study brain function in patients with schizophrenia, we have been doing this for the past 30 years, so we know that there is some differences between patients and controls. So how do we bring this information from post-mortem to in vivo phenotypes? By using a polygenic co-expression index, we use a very complicated way to calculate this thing, but basically what we do is that we characterize the genetic variation predicting the co-expression of a set of genes as analyzed with post-mortem. So with that number, the polygenic co-expression index, we can perform associations and correlations with in vivo imaging phenotypes or other phenotypes. And the imaging phenotype we have been working for the past 30 years is working memory cortical inefficiency, which we and others around the world have demonstrated is a main characteristic of how the brain in patients with schizophrenia processes information in an inefficient way. And from animal studies of Patricia Goldman-Rakich, for example, it has been shown clearly that the dopamine D1 and dopamine D2 receptor contribute significantly to determine efficiency of brain processing, especially during working memory. So this is the first example. This is just a graph showing what we did, which is summarized in these two papers here by Giulio Pergola and Pierluigi Selvaggi, published a couple of years ago. And in this first study, we performed the method, we established the method, and then what we did, we characterized a set of genes which is co-expressed with the dopamine D2 receptor from a microarray study of the dorsolateral prefrontal cortex from post-mortem human brain. We calculated the polygenic co-expression index, and what we did then, we used the polygenic co-expression index to perform the association with human brain activity in vivo, of course, in healthy subjects. You see, we obtained two independent replications. This is a sample from the Lieber Institute for Brain Development again. So two independent, one independent replication of our results, showing that the greater the polygenic co-expression index, namely the greater the co-expression of the whole set of genes being co-expressed with the dopamine D2 receptor, the greater the activity of the dorsolateral prefrontal cortex during performance of the NBAC working memory task, which is consistent with what has been shown with PET studies and fMRI studies before. by the group of Slifstin and others a few years back. Also, recently we also published another paper in PNAS in which Leonardo Fazio, Giulio Pergola and Giuseppe Blasi from my lab performed a similar study studying the set of genes being co-expressed with the dopamine D1 receptor, and again we have a discovery and replication analysis in which again there is a similar relationship between the polygenic co-expression index of the dopamine D1 set of genes with cortical activity during the end-back working memory task, and you see that the direction of the correlation this time is negative, suggesting that the more co-expressed are these genes, the more efficient is the cortical activity during working memory. Finally, paper which is coming out in biological psychiatry, what the next step, of course, we wanted to try and evaluate is whether any of these sets of genes as identified with network analysis is enriched for genetic risk for schizophrenia. Long story short, Giulio and Pasquale Di Carlo identified a set of genes which is highly enriched of genes that are known to be involved in risk for schizophrenia. For those who know something about the genetics of schizophrenia, you see that several of these genes have long been known to be involved in genetic risk for schizophrenia, like CACNA1C, GRM5. So once we identify this module which is enriched for schizophrenia genes, and this is actually replicated across 5 different samples, we want to see whether there is any relationship of this module of co-expressed genes, so a biological pathway which we, in a data-driven fashion, we have identified, whether there is any correlation with, for example, something which is very important to any psychiatrist in the world, which is response to antipsychotics, in this case from the Katie trial and from our own sample, independent replication again, in which we show that there is a significant correlation, that the higher is the co-expression of these genes, the lower is the response to the antipsychotic treatment. I think I've been very on time. Just some acknowledgements before quick conclusions. I would like to acknowledge the collaborators at the Liber Institute for Brain Development, like Luca Orsini, Anand Mattei, and Daniel Weinberger, who also happens to be my mentor, and then Fabio Sambataro at the University of Udine, and the PIs in my group, like Giulio Pergola, Giuseppe Blasi, Grazia Caforio, Antonio Rampino, and Linda Antonucci. Just 1 second for very simple and plain conclusions. Identification of genetic variants associated with schizophrenia is ongoing, so we are at a good stage with this, but we need to understand how this genetic risk, these genomic loci, create biological risk for schizophrenia. The interaction between genetic risk and environment is something that we need to keep into account, and we believe we are using a tool like gene networks, genetic networks, to identify biological pathways which translate genomic risk for schizophrenia into in vivo phenotypes, which we can also measure. Thank you for your attention.
Thank you, Alessandro.